Eko Susanto, MSc. Time is yours.
Nục rửa gọi chi lý. Đấy.
Honorable Rector of the University of Lampung, Dr. Muhammad Sufyan Effendi, MED, Dean of Faculty Mathematics and Natural Sciences, Dr. Eng Suripto Dwiyono, MT, Head of LPPM and also Head of LP3M, the University of Lampung, distinguished participants, ladies and gentlemen, good morning. Tabi pun, I'm honored and delighted on behalf of the conference committee to welcome you to the 4th International Conference on Applied Science, Mathematics and Informatics 2022 of the Faculty of Mathematics and Natural Sciences, University of Lampung. The theme of the conference is the role of natural science, mathematics, and informatics for the development of advanced material based on natural resources, to highlight the part of science in handling issues and making synergies with other areas on the economical valorization of natural resources. Even in the post-pandemic of COVID-19, the ICASMI is held virtually on September 8 and 9, 2022 in Bandar Lampung, Indonesia. The ICASMI allow researchers, scientists, and also representative of industry, government, employer, and postgraduate students to have an opportunity to share their works and idea in this precious time. We wish to take this opportunity to welcome all participants, the keynote speakers, and our invited speakers for coming to our campus, the University of Lampung, virtually. Ladies and gentlemen, the conference consisted of 3 sessions: plenary, parallel session, and panel presentation. The plenary session has 4 keynote speakers who are competent on their field of study, come from different countries: Pakistan, Malaysia, Thailand, and Russia, that will deliver their speech. The parallel session will divide it into 5 parallel in different fields of study: biology, chemistry, mathematics, informatics, and physics. And every session will have 2 invited speakers. And finally, the panel presentation will have all of the oral presenters who will talk for 10 minutes in each class. The number of registered participants is 236 participants, consisting of 183 oral presenters and 53 non-presenters who come from more than 20 university and research institutions in Indonesia, and also some participants from abroad university. Ladies and gentlemen, our technical program is rich and varied with keynote speakers, invited speakers, and 7 parallel sessions. On this occasion, I also thank to our keynote speakers. First, Associate Professor Chatchawan Chanta Suryat from Kasetsat University, Thailand. Second, Associate Professor Muhammad Muazzam Nazar from Quaid-Azam University, Pakistan. Third, Associate Professor Adi Baswib from University Teknologi Mara, Malaysia. And also Professor Alexander Briaskin from Batker Institute of Nuclear Russia. And also, we are talking to invited speaker, Professor Agus Sugianto from Erlangga University, Mrs. Niluh Watiniasih PhD from Udayana University, Professor Dr. Hermansyah from Sriwijaya University, and Professor Dr. Rudi Stumiang from University of Lampung. Professor Inengah Suparta from Ganesha University of Education, Professor Dr. La Zakaria from University of Lampung, Professor Dr. Hoga Sarage from Bakri University, and Dr. Samsuryadi from Sriwijaya University, and also to Dr. Ing Idris Mandang from Mulawaran University, and Mrs. Dwi Asmi, PhD from University of Lampung. Ladies and gentlemen, I would like to report that the selected paper presented at this conference will be published in American Institute of Physics IP conference proceeding, indexed in the leading database such as Web of Science, Scopus, Inspect, Chemical Abstract Service, and Astrophysics Data System. Finally, as the chairman of ICASMI 2022, I know that the conference success depends ultimately on the many people who have worked with us to plan and organize both the technical and supporting arrangements. I thank to the organizing committee members who have all worked extremely hard to detail essential aspects of the conference programs, including Director of University of Lampung and the Dean of the Faculty of Mathematics and Natural Sciences. I hope this conference is able to inspire and deliver benefits to all participants, in which together we are able to contribute to science and research. Thank you very much for your attention, and I hope you will enjoy this conference. Thank you.
Thank you very much, Dr. G. Nugroho Susanto, MSc, for the report.
Next, we will hear the welcome speech by the Dean of Faculty of Mathematics and Natural Sciences, University of Lampung. For Dr. Engineering Sripto Dwi Yuwono MT, time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Rector Lampung University, Dean at Lampung University, the head of LPPM and the head of LPPPM, the all keynote speaker, invited speaker, and all participants. In the name of Allah, the most beneficent, the most merciful. Tabi pun. It is my great honor on behalf of the Faculty of Mathematics and Natural Science to welcome all participants to the 4th International Conference on Applied Science, Mathematics, and Informatics. The theme of the conference is the role of natural science, mathematics, and informatics for the development of advanced methods based on the natural resources. even though in the post-pandemic COVID-19, we are still able to organize this conference. We hope this conference can be platform together the disseminate a new innovation and research in science, in particularly natural science, applied science, mathematics, and also computer science. Researchers, academicians, and the students are able to share and discuss a new finding, the application of the science. The aim is to invite collaboration between academics, researcher, and industry, both national and international. As started in conference, we encourage innovation and research for development of advanced material and natural science. One of the popular based on natural science and natural resources. Ladies and gentlemen, of advanced material based on natural resource are one of the popular in my faculty, Lampung University. Currently, the faculty Mathematics and Natural Science has more than 3,000 students spread across in 12 study program. We have one diploma program, 6 undergraduate program, 4 master program, and 1 doctor program. Our faculty member also includes 18 professors and 1 more 87 assistant and associate professor. Therefore, with faculty, this resource, there are many opportunity to conduct collaboration with our faculty. Besides international conference, our faculty also periodically organize a national conference at we call SMIAP. We have held this conference regularly also. Thank you, Director of the University of Lampung, Head of LPPM, Dean, Head of LP3M, and all the keynote speaker, invited speaker, and all participants. I'd also like to give my gratitude to the organizing committee for their hard effort in organizing the conference. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Dr. Ang Sripto Dwi Yuwono, for the welcome speech.
Lastly, we will hear the welcome speech by the Rector of University of Lampung. And also, we'd like to have his honor to officially open this event. And his welcome speech has been recorded by the committee. For operator, please play the video.
Assalamualaikum warahmatullahi wabarakatuh. Tabik pun, may the peace, mercy, and blessing of Allah be upon you. Distinguished guests, speakers, participants, ladies and gentlemen, praise and gratitude to Allah Subhanahu wa Ta'ala, the Almighty God. Because of His grace and guidance, we can attend the opening ceremony of the International Conference on Applied Science, Mathematics, and Informatics 2022. The theme of ICASMI is the role of natural sciences, mathematics, and informatics for the development of advanced material based on the natural resources. It aims to highlight the role of sciences in the tracking problem and creating synergies with other fields on the sustainable valorization of natural resources. This is one of the clear evidence that the University of Lampung has a strong commitment to contribute positively to the development of advanced material based on the natural resources, especially in Lampung province. Ladies and gentlemen, advanced materials are known to have the most improved popularity in modern civilization and considered as a crucial element toward the economic growth and well-being of the society. Advanced materials have the probability to contribute to the sustainable development goals in the different ways. The fabrication of advanced material has a particular ecological and social importance. Their use in modern teach application, healthcare, energy sector, environmental importance, etc. reveal their important role for economic and sustainable development towards society well-being. Through this event, the University of Lampung, which is located in the southern tip of Sumatra Island, Indonesia, take the initiative to facilitate this by organizing the ke-4 ICASMI, and it will be provide the opportunity to the researchers, sciences, and students from sciences, mathematics, and informatics, also from various application areas such as life sciences, agriculture, and engineering, to share their work, thoughts, ideas, learn from and lean on each other in this challenging time. Ladies and gentlemen, before we begin this conference, I would like to express my gratitude to the organizing committee who sincerely committed to this event to make it a success. Also to our keynote speakers who are willing to present and share their expertise in this conference. Finally, as Director of the University of Lampung, I hereby declare the conference officially open. Have a great conference to all of you, and hopefully our participants in this event can be our contribution to the community, and may Allah bless our noble efforts. Billahi taufiq wal hidayah. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Dr. Muhammad Sofwan FND MED, for officially open this event.
Before we end this opening ceremony, let us say a prayer that will be guided by Dr. Saiful Bahri, MSI. For Dr. Saiful Bahri, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, let us bow our heads for a moment, pray to God, Allah subhanahu wa ta'ala, so that our international conference can be praises to all of us. I will lead this prayer based on the teaching of Islam, and those who are not Muslims, you are pleased to pray according to each of your beliefs. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. الحمد لله رب العالمين حمدا يوافي نعمه ويكافئ مزيده. يا ربنا لك الحمد كما ينبغي لجلال وجهك الكريم وعظيم سلطانك. اللهم إنا نسألك سلامة في ديننا وعافية في جسدنا وزيادة في علمنا وبركة في رزقنا وتوبة قبل الموت ورحمة عند الموت ومغفرة بعد الموت. اللهم حوّن لنا في سكرة الموت ونجاة من النار والعافية عند الحساب. Oh Allah, today, Thursday, September 8th, 2022, in this beautiful place, we gather here to bring an international conference on applied science, mathematics, and informatics. With the theme is the role of natural sciences, mathematics, and informatics for development of advanced material based on natural resources. Make this international conference is a useful science assembly as a medium of sharing useful ideas, knowledge, and experience of scholars, researchers, and students of various disciplines. May the conference we organize today benefit to our lives, broaden our knowledge, shine our ideas, and lead us to be successful, productive persons, which in turn will boost dignity of our nation. Oh Allah, guide and bless our hearts and our minds with the lights of your guidance. Empower your supreme wisdom upon our activities. Help us to speak our minds clearly. Help us to listen to each other, respect each other, love each other, so that we are included to the blessed persons. Oh Allah, protect us from unintended temptation. Show us the right path and give us strength from good things. Equally, show us that the bad things and give us knowledge and strength to avoid them. Oh Allah, you are the one who can fulfill our dua. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzaban nar. Subhana rabbika rabbil izzati amma yasifun wa salamun alal mursalin walhamdulillahi rabbil alamin. Assalamualaikum warahmatullahi wabarakatuh.
Amin. May all God blessings come to us. Thank you very much, Dr. Saiful Bahri.
Ladies and gentlemen, now we come to the end of the opening ceremony of
The fourth International Conference on Applied Science, Mathematics, and Informatics. For closing this opening ceremony, let us say Hamdallah.
Next agenda is photo session. For all the participants may turn on the camera.
Okay, for the first slide. Okay, the second slide. 1, 2, and 3. Okay, The first slide, 1, 2, and 3. The second slide, 1, 2, and 3. Okay, that's all. Now we will move to the main event, the keynote session. For the first keynote session, we'll be guided by Endang Linirin Widyastuti, PhD. But before that, let me read her brief curriculum vitae. Okay, for our first moderator, her full name is Endang Linirin Widyastuti, PhD. She is one of Associate Professor in Department of Biology, Faculty of Mathematics and Natural Sciences, University of Lampung. Her educational background, the Bachelor, Indonesian University of Education, Department of Biology in 1985. For master degree in the University of Kentucky, Biological of Science, 1991. And doctoral in the same university, University of Kentucky, Biological of Science, 1997. And her publication, there are 20 publications in the last 5 years. One of them in the 2022, Marine Space Allocation Analysis for Coastal Area Small Island Zone in Lampung Province. Please welcome our first moderator, Endang Linirin Widyastuti, PhD.
Okay, thank you very much for very fine introduction. Ladies and gentlemen, maybe we can start with the first speaker that is going to give by Dr. Cacawan Janta Suri Jarat. I hope that I'm not misspelling your name. Dr. Janta Suri Jayat is born in 15 April 1976, and he's an associate professor right now. He's working in the genetic department of Faculty Science, Kasasat University in Bangkok. His background, he has an additional background as a bachelor degree from Kasas University, and he got a master degree in the crop science of the Oregon State University, and he got the PhD also in a pathology from the Ohio State University. Is anyone, is my voice can be heard clearly?
Clear.
Okay, thank you, Prof. Okay, I will continue again. A lot honor and award from Asia Unit Austria's Staff Exchange in 20, and he has a Newton Fellowship for the Mid-Career Development Program in 2017 also. He also has Erasmus Mundus Expert Program Staff Exchange in Croatia in 2015. He also has a Fulbright Honor Award from US as Asian Scientist Fellowship in 2013 for gift and contribution of Dr. Janta Surijayat is a committee of the Society of, so he is an editorial board member of Agriculture and Natural Resources, and also he's a editorial board for Maritime Science 2015 until the present. And he has a lot of things also with the publication, especially he has more than 100 publications, and especially the recent publication is about genetic distribution of the virulence gene, FVPS, In Thailand, rice blast solid and to the broad spectrum resistant rice variety. But in order to make it short, because we need more information from you, Dr. Janta Suryajaya. So you have 20 minutes to deliver your talk. Time is yours, please.
Thank you very much for the introductions. First of all, I would like to thank the organizing committee of this conference that give me the opportunities to present my research work at ke-4 universities to all. So I will start with sharing the screen of my presentation. So can you see my slide?
Yes.
Okay, so
It's clear.
So the topics that I'm going to talk about today is the research work that has been carried on in my lab during the last 15 years, which is the genetic diversity and molecular interaction between rice and rice blast fungus in Thailand. So I believe that all of us agree that rice is one of the most important crop worldwide, especially for ASEAN countries and Asia countries that we all eat rice every day. And many countries in these regions are the exporter of rice. And Thailand is actually the world #2 rice exporters. Each year we export more than 2.8 million tons. And several rice varieties from Thailand are very famous, especially for jasmine rice and the RD6, the sticky rice version of the jasmine rice. But each year, you know that there are many factors that affecting the rice productions, both the biotic stress and the abiotic stress. And one of the biotic stress that's really important for the rice production is the rice blast disease, which caused by Magnapothe oryzae, the fungal pathogens. And these figures showing you the normal or regular symptoms of the rice blast disease that's showing on the leaf. And each year, the rice production has been affected by this disease, about 10 to 30% of yield loss. So in order to protect our rice production and rice yield from this disease, we need to understand the disease cycles of this fungus. So basically, the asexual spore, the conidia of the rice blast fungus, can blow by the wind or splash by water and then landed on the rice leaf surface. And then it will start to penetrate into the rice tissues and colonize inside the leaf tissues, and then start to produce more spores and then repeat these disease cycles. And then the disease will spread out really quickly, like basically every 7 days, then it can repeat the whole cycles of this disease. So once the rice field has been infected with the disease, then the whole rice field can be infected very quickly. So in general, there are many strategies that farmers has been used to control or to protect the rice farm with for this disease, including the adjusting the planting time to escape from the heavy infection of the fungus, or do the crop rotations by using the non-host plant species to grow in the field, or reduce the amount of the nitrogen fertilizers, which is the favor of the fungal growth. And of course, the major strategies that most farmers use is the chemical fungicides, which can effectively control the disease, but it's not environmental friendly, and also increase the cost of growing rice. So, yes?
Ganti. Okay.
Okay. Okay, and the best way that the breeders and the scientists recommend the farmers to do is to use the resistant rice varieties, which is the economical and environmentally friendly to the world. So in order to have the effective rice resistant varieties, we need to understand the basic concept of the gene for gene theories, which saying that in rice plants, the host plants has the resistant genes, and in the pathogens, the fungus has the AVR genes. And if the resistant gene from the plants can recognize the AVR or the effector genes from the fungus, then rice plant will trigger the defense response and protect the rice from getting the disease. But in other cases, if the rice doesn't have the resistant genes or the fungus doesn't produce or produce different kinds of the AVR proteins or effectors, then basically the rice plant will not know that the fungal is present, and then the fungal will grow and the rice plant will get the disease. So from these concepts, in the past 30 years also, there are many resistant genes from rice and many AVR proteins from fungus has been cloned and characterized. And in this slide showing 27 genes that has been cloned from the resistant proteins and 11 AVR genes that has been cloned and characterized. So in my lab and my collaborator in Thailand, we have been collecting the rice germplasms all over the countries and then screening for the broad resistance spectrums to identify the rice varieties that can perform well in Thailand. And we found that one of the rice varieties named Chao Hom Nin, or from now on, I will use the abbreviation of the JHN, showing a very broad spectrum to a wide range of the rice blast fungus isolates in Thailand. So our colleagues in Thailand here, using the QTL mapping technique to identify the location of these resistant genes in Chao Hom Nin rice varieties, and we narrowed down it to 2 locations in the rice genome, one on chromosome 1 and one on chromosome 11. And then later on in my lab, one of my students spending some time to identify and clone these genes and showing that the reason that JHN is showing a broad spectrum resistance is because it contains 2 very important resistant genes to rice blast fungus. The one on chromosome 1 is PISH, and another one on chromosome 11 is PIK. So with that, we would like to understand if the defense mechanisms of these 2 resistant genes, the PISH and PIK, is sharing the common signaling pathway, or do they have the specific signaling pathway for each of these 2 genes? So we perform the transcriptome analysis by making the near isogenic lines of the rice that contain individual PISS genes or PIK genes, and also the ones that has both genes in the same background, and then use the transcriptomic technique or RNA-seq to identify the genes that is responsible for the resistance of these 2 resistant genes. And then not only we focusing on the plant site or the host site, we also need to understand how the fungus is adapting or living in the environment. So we also collect the rice leaf in the field around the countries and then bring it back to the lab and then isolate the fungus out from the rice leaf that showed lesions of this disease. And then we characterize both morphologically and using the DNA markers to characterize all these rice blast fungus populations using the ISSR and SRAP or SLAP markers. So this slide showing an example of the morphology of the rice blast fungus isolate that we collect from all over the countries. And then the bottom portion of this slide showing the very diverse shape of the conidia that these rice blast isolate produce. Then we perform the DNA marker analysis to study the genetic diversities and relationship of this isolate. You can clearly see that rice blast isolate population in Thailand show a very diverse and very high genetic level of the differentiations. So to further analysis of the genetic diversities of the rice blast fungus population in Thailand, 2 of my students are focusing on the sequence of the AVR genes in the fungus. So we select 3 of the most important AVR genes, at least for Thailand, which is the AVR PI9, AVR PIK, and AVR PITA1. So we amplify the genomic sequence of these 3 AVR genes, sequence it, and then use this sequence to analyze for the genetic diversity. And interestingly, we found that not all 3 showing the same pattern of the genetic variations. For example, the AVR PI9, it is very conserved in pretty much 100% or 99% of the rice blast fungus isolate in Thailand, that they all have these genes with no genetic variations. And when we look at the literatures from the researchers in other countries, we also found the same thing, that in China, in Japan, in Korea, in Philippines, all the rice blast fungus, or the majority of the rice blast fungus, do contain these AVRPI9 genes. So somehow this gene is very important for the fungus, so that they keep this gene in all the fungal isolate from around the world. On the other hand, for AVRPIK and AVRPTA, we see a very high genetic variations within these two genes. So there are several alleles and there are many mutations and transposon insertions into these two genes. So from this study, we conclude that different genes, the AVR genes, do have different level of the genetic variations. So next, we would like to study further about the AVR-PIK genes, because the PIK is found in JHN. So that we sequence these AVR-PIK again from the rice blast fungus in Thailand. And surprisingly, we found that instead of having only one copies, some of the rice blast isolates in Thailand do have 2 copies of these AVR-PIK genes. And we analyze the sequence of these AVR-PIK genes, and we show that in general, in the past, it has been 5 alleles that has been reported for these AVR-PIK alleles, the AVR-PIK A, B, C, D, and E. And these 5 alleles only difference at 5 amino acid positions of the 113 amino acid polypeptide chain. And then in our study, we identified the novel AVR-PIK alleles, and we named it as the AVR-PIKF. And this AVR-PIKF showing a very interesting characteristic, that is only show 1 nucleotide difference from the AVR-PIK-A alleles that has been reported earlier. And when we check the pathogenicities of different AVR-PIK, the AVR-PIK-A, C, D, E, and F, and also those isolates that contain 2 copies of this AVR-PIK. So I want to make it clear at the beginning that the isolate that contain 2 copies is always contain copy of the AVR-PIK-D and the novel alleles of the AVR-PIK-F. So when we check the disease with the JHN rice, we found that JHN rice varieties can show the resistant reaction to only rice blast fungus that contain AVR-PIK-D, but it cannot protect the rice plant from the rice blast fungus that contain other alleles. For example, AVR-PIK-A, C, E, and of course, the novel alleles, the AVR-PIK-F. And another interesting thing is that when the rice blast isolate contain 2 alleles, and one of the allele is the allele D, then the JHN rice can protect the rice from getting the disease. And we use jasmine rice or cow dog millet rice as the negative or the susceptible control. So next, we check how bad or how aggressive of the AVR-PIKF alleles by inoculate these AVR-PIKF rice blast fungus to different rice that contain different AVR-PIK alleles. In nature, we have 7 or 8 different AVR-PIK alleles in rice. As you can see here, this is the rice plants that contain PIKA, the rice plants contain PIKD, PIKM, PIKH, PI1, PI7, PIKS. These are all the alleles, different alleles of the PI resistant gene in rice. You can see that the rice blast fungus that contain AVR-PIKF, the novel alleles that we just identified, show the very aggressive phenotype to all the existing resistant PIK alleles in nature. So basically, there is no resistant alleles that can recognize and protect the rice from these AVR-PIKF alleles from the fungus. And then on the right-hand side, we validate the binding of the resistant proteins, the PIK, and the effector or AVR proteins from the fungus. We show that if the rice plants can be protected by the fungus, it means that the resistant protein can bind to the effector protein from the fungus. So in these figures, we use the gel infiltration chromatography. So once these two proteins can bind, then the complex get bigger and it will come out from the column earlier. On the other hand, with the AVR-PIKF, the novel alleles, it cannot bind to the resistant protein, so that you will not see the big complex that come out earlier in the chromatography columns. And we also show using the Nicotiana benzamela that if the resistant proteins, the PIK, can recognize the effector protein from the fungus, for example, the AVR-PIKD, then the Nicotiana benzamela will show cell death. But on the other hand, when we infiltrate resistant protein together with the AVR-PIKF, and then we know already that they cannot bind to each other, then you will not observe the cell death. And this is another experiment to confirm that the resistant protein and effector protein from the fungus can bind and can trigger the cell death or defense response in rice. So after we know that we identify the novel alleles on AVR-PIK, then we also would like to understand how these AVR or effector genes has been regulated by the fungus. So we characterize the promoter of these AVR genes by using the AVR-PI9 as the model. So we download the sequence of several isolates and then identify the regions of the AVR-PI9 promoters, clone the sequence, and then construct the truncated promoter sequence to analyze what is the minimum sequence that is still function for these promoters by fuse these fragments of the promoter together with the GFP reporter genes and then transform into the rice blast fungal protoplast and then use these transgenic fungus that has the promoter of the AVR-PI9 with the GFP reporter genes to infect rice plants using the leaf sheath inoculations. And we found that if the fragment of the promoter is shorter than 700 base pair, you will not see the green fluorescent signal. But if the promoter fragment is 900 base pair, then you can see the green fluorescent signals. So basically, the 200 base pair between 700 to 900 is very important region of the promoter for the function of the AVR-PI9. So we further characterize by using the DNA protein pull-down assays by using that 200 base pair fragments to fish out the protein from the fungus, trying to identify the transcription factors that bind to these 200 base pair regions. We actually obtain several candidate proteins, but one of the interesting proteins showing to be the transcription factors, the Mohawk 6. So we are at the current status trying to validate and confirm how the Mohawk 6 can regulate these AVR-PI9 genes. Another thing is that we're trying to identify the target proteins or the rice target protein of the effector AVR-PI9. So once the fungus deliver these AVR-PI9 effectors into the rice cells, what is the target protein in the rice cells? So we do the protein pull down by expressing the AVR-PI9 proteins and then incubate together with the protein extract from infected rice and healthy rice, and then go to the columns and then use mass spectrophotometer to identify the candidate proteins. So at the current research status, we're trying to validate the binding between the AVR-PI9 promoters and the transcription factors from the fungus using gel chip assays or EMSA. And we're also trying to validate the binding between the AVR-PI9 effectors and the target protein in rice using these two hybrid assays. And we also trying to using the new technique, which is called the single cell transcriptome analysis, to identify the gene expression patterns of the rice that infected with the rice blast fungus. The important concept of this one is that we infect the rice with the fungus, then we isolate the protoplast from this infected rice and use the cell sorting machine to deliver each individual cells into the droplet of the oil or emulsion oil. And then each individual cell will be used to perform the library constructions and then subjected to the RNA seeds. So basically we'll perform the transcriptome for each individual cell. So we will be able to obtain the expression patterns of each individual cells and would be able to understand the defense mechanism in rice plants better than before. So in the last research experiment that I would like to tell you is by developing the identification techniques. In the past, we have to grow the plants, culture the fungus and spray and then check the symptoms, or we have to isolate the DNA and do the PCR amplification and sequence in order to identify the alleles or the effector genes from the fungus, which takes several budget and time and from the technicians. So we're trying to apply the CRISPR-Cas12a system to develop the identification kit of the AVR genes and alleles. With the activities of Cas12a enzymes, which has the collateral cleavage, which means that once the guide RNA is already triggered, these Cas12a can non-specifically cleave the single-stranded DNA. And we use this characteristic to develop the detection kit for the AVR genes. So first, we use the AVR PIK as the model because it has several alleles, A to F. We design the specific guide RNA for each of the alleles. And then we use the fluorescent-based detection assays because it's easy for the farmers or the breeders to check in the fields or in the very classical lab. So once the guide RNA is triggered by the specific target DNA, then the probe, which is labeled with the fluorescent, will be digested. And then it will trigger the clean fluorescent colors, and then we can observe these signals by using the LED transilluminator. And this is the preliminary results that we obtained from different guide RNA, and we can see that there are still some cross-contamination of different AVR-PIK alleles. So we are in the process of optimizing the conditions so that we can specifically using this technique to identify different alleles from the rice blast fungus. And in the future, we also have planned to use this same technique to modify it into the lateral flow detection assays or the strip test, just like we use the strip test, the ATK test for the COVID-19. So basically, if the target DNA is triggered by the guide RNA, then the Cas12a will digest the probe and then release the biotin, and FAM will move along the lateral flow strip and then get stuck with the antibodies and show the second signal strips as the positive there. So with that and the time that I have, so I would like to thank my collaborators from Seoul National University, Professor Young Hwantney, and from UK, Joint in the Center, Professor Mark Banfield, and my collaborator in Thailand from Camille, Dr. Nolak Parentwong, and from Faculty of Agriculture, Kasetsat University, Dr. Thani Sibongchai. So with that, thank you very much for your attention.
Thank you. Thank you very much. It's very insightful about the plant pathology. Anyway, maybe we have run 2 minutes, but it's okay because your material is very, very interesting. Okay, we come up with the second keynote speaker from Dr. Muhammad Muazzam Nasser. Dr. Nasser, are you there?
Yes, I am.
Okay, great. Okay, great. Let me introduce your CV first to the audience. Dr. Muhammad Muazzam Nasser is hold on a second. Okay. So he's born in Chakwal, Punjab, Pakistan in 9 October 1979. And he has the education for PhD program in organic chemistry from the Institute of Chemistry, Chinese Academy of Science, Beijing. He got the 2 master degree, Master of Philosophy in Organic Chemistry from the Bahaddin Zakaria University, Pakistan. And also he got a master degree from University of Agriculture, Faisal Abad, Pakistan. As well as Dr. Chazawat, Dr. Nazir also published more than 100 publications, and one of them that the most recently is a let me, because I have a pile of your CV, Dr. Nazir, so I have to flip over all of the paper about your publication. Okay, so the most recently publication is about molecular duplex featuring the NHH2, CHO, and CH interaction in a solid state self-assembly of triazine-based compound. Dr. Nazir also have an award and honors for many different agency, particularly from German and also from New Zealand and so on. Professional experience of Dr. He is an associate professor in the Department of Chemistry, Quaid Azam University, Islamabad, Pakistan. He also worked as assistant professor in the Department of Chemistry, and also he worked as a lecturer in in the same department of the chemistry from the same university, and also now he's a associate professor from the Department of Chemistry, Quaid Azam University in Islamabad, Pakistan. I would like to introduce about the paper or the talk that he is going to give a talk for today. Is isotin dioxide indole as a privileged nucleus in the medicinal chemistry. I would like to take more time. I give you time to deliver your talk, and you have 20 minutes for deliver your talk. The time is yours, Dr. Nazir, please.
Thank you very much, Professor, for your kind introduction. Just give me 1 minute. Yes. Okay. Today I would like to tell you about isotin. Is a chemical moiety that is a kind of privileged nucleus in medicinal chemistry. So we have been working on this nucleus for the last 8 to 10 years. So mainly I will focus on the work we have done in the past 2 to 3 years. First, I would like to introduce this moiety to you. The isotin is a well-known natural product which can be found in the plants of genus Isotis and many other plants. This indolic derivative has been known in the medicinal industry for nearly 140 years, as it is a product of indigo dye oxidation by means of nitric and chromic acid. It is an endogenous oxidized indole present in the mammalian brain, peripheral tissues, and body fluids. Isotin has been extensively studied in behavioral and metabolic assays, and more than 90% and more than 90 proteins have been identified through proteomic analysis as its potential biological targets. It is a flexible and versatile synthetic scaffold consisting of an indole nucleus, two types of carbonyl groups, a keto and a lactam group. If you look at the structure of the iCETIN, there are three different reactive functional groups, which you can easily modify to make the derivatives of this. The first is the C3 carbonyl. If you look at this carbonyl, keto carbonyl, this is C3 carbonyl. So you can have the different reactions on this carbonyl. You can make different hydrozones, shift bases, pyro compounds, thiosammicarbozones, etc. There are a lot of derivatives have been prepared by using the reactivity of this 3-carbonyl. Further, you can have the substitutions at the different substitutions at this phenyl ring, and you can have mono, di, tri substitutions, and there are a lot of derivatives. Now there are many derivatives that are commercially available having different substitutions at this aryl moiety. The third important site is the N substitution. You can have because this N can act as nucleophile, you can have different alkyls, acyls, and aryl substitutions to prepare different derivatives. So out of many derivatives, thousands of derivatives of iCETIN, so they have been explored in medicinal chemistry. They are reported to have antibacterial activity, antifungal activity, anti-cancer, anti-tubercular, anti-diabetic, anti-HIV, and many more activities. You name the activity and the iCETIN has that activity. So this is very important moiety in medicinal chemistry. If you look at this slide, there are a number of FDA approved drugs, and that has iCETIN moiety in their structures. And there are some of the iCETIN based drugs that are presently in clinical trials. For example, this is sunitinib is an anti-cancer drug currently used are available in the market. So why this iCETIN is so important in medicinal chemistry? Because of its structural features. It has the capability of making hydrogen bonds. So it has 2 hydrogen bond acceptors in the form of the keto carbonyl and the amidic carbonyls. The presence of 2 highly electronegative oxygen atoms renders the 5-membered ring of iCETIN an electron deficient ring. Electron deficient ring offering an interesting duality to the iCETIN that already has fused electron rich 6-member ring. So this 6-member ring is electron rich ring, and the 5-member ring is electron deficient ring due to the presence of 2 nearby oxygen atoms. So owing to this special structural feature, iCETIN moiety has high capability of making strong anti-parallel pi-pi stacking interactions. So you can see in this pictures the formation of anti-parallel pi-pi stacking interactions. The most recently explored carbonyl-carbonyl interactions. So you can also observe the structural features in this iCETIN moiety that can make the carbonyl-carbonyl interactions as well. So due to these interactions, this iCETIN moiety is very important, and with the help of these interactions, it can interact with the target binding sites. So that's why this is very important. So as we all know, worldwide, the prevalence of chronic non-communicable diseases is increasing at an alarming rate and approaching epidemic proportions. About 18 million people die every year from cardiovascular and neurodegenerative diseases, for which diabetes is the major predisposing factors. Diabetes is a chronic metabolic disease with high blood glucose levels that can damage blood vessels and nerves and causes multiple serious complications. According to the International Diabetes Federation, 1 in 11 adults had diabetes, and about 425 million people are suffering from diabetes mellitus. And 12% of the global health expenditure was spent on diabetes in 2017. Diabetes mellitus is a chronic metabolic disorder characterized by hyperglycemia resulting from lack of insulin secretion, impaired insulin sensitivity, or both. Diabetes mellitus can be divided into two main categories, insulin dependent and non-insulin dependent. Type 1 DM is associated with the absolute functionality loss of the human pancreatic beta cells to produce insulin, a hormone that regulates glucose. Whereas type 2 diabetes mellitus is related to the insulin resistance as well as the high glucose and insulin levels at the first stages of the disease. One of the options to control this hyperglycemia in diabetic patients is to inhibit the digestion of dietary carbohydrates. The enzyme alpha glucosidase in small intestine is responsible for the hydrolysis of dietary carbohydrates such as starch and disaccharides to glucose that enters the bloodstream, resulting in the hyperglycemia. Consequently, the inhibition of alpha glucosidase can reduce blood sugar levels by suppressing carbohydrate digestion. Presently, acarbose, voglibose, and migilitol are commercially available alpha glucosidase inhibitors, but unfortunately, they are reported to have serious adverse side effects like diarrhea, abdominal discomfort, bloating, etc. Hence, novel and more effective alpha glucosidase inhibitors are presently much needed. So current research suggests that hybrid molecular approach, which involves the combination of two molecules with individual intrinsic activity into a single agent, is an effective way of producing more effective compound. This strategy has been found superior to the conventional combination therapy. Advantages of this hybridization approach are development of new drugs to overcome the cross resistance may result into a compound with enhanced potency, multiple modes of action, and reduced side effects. So, keeping this in view, we designed many isoden-based compounds and studied the alpha glucosidase inhibition activity of those synthesized compounds. Now I will tell you systematically about the synthesis of those organic compounds which we synthesized in our laboratory and tested them for alpha glucosidase activity to control the diabetes mellitus. So as I introduced on the previous slides, that hybridization approach is useful approach to produce more effective agents. So we try to hybridize this isoden moiety with other pharmacologically important and potent heterocyclic moieties. So our first scheme was the synthesis of phenoxypennate isotenes. We reacted isoten with bromoethoxy substituted phenols in the presence of potassium carbonate and DMF solvent at 50 to 60 degrees centigrade for 3 to 4 hours to produce this the final phenoxypennate isotenes. So if you look at the physical data of these phenoxypennate isotenes, we will observe these all were obtained in good to excellent yields. So here are the alpha glucosidase inhibition activity of these phenoxyphenate isoteins. And you will observe that as compared to the standard positive control acarbose, our compounds were found to be much more active. Next was to condense and to use the reactivity of 3 carbonyls. So the different hydrazides which we prepared in our laboratory were condensed with the phenoxyphenate isoteins in ethanol solvent under flux conditions in about 2 to 8 hours to get the final hydrazones of phenoxyphenate isoteins. So this table shows the yield and the melting points. If you observe the yields, these were also produced in good to excellent yields. So here is the activity, alpha glucosidase inhibition activity, and we found that the activity is exceptional for these hydrazones. It is slightly better than the phenoxyphenate isoteins. But still you can observe that all the compounds are much more active as compared to the standard acarbose. Next, by utilizing the same 3 carbonyl reactivity, we treated this phenoxyphenate isoteins with thiosemicarbazides and produced the thiosemicarbazones of phenoxyphenate isoteins in good to excellent yields. Here are the activity table, and you can see that the activity was further improved. And if you compare the hydrazones with the thiosemicarbazones, the activity is slightly better. But still, if you compare with the acarbose, these compounds are highly potent compounds. So we were very happy to get these compounds. And the next step was to further derivatize this and to produce the heterocyclic moiety thiazole on the framework. So we treated this thiosemicarbazones of phenoxyphenate isoteins with alpha halo carbonyl compounds in the presence of catalytic amount of acetic acid in ethanol under flux to produce the desired thiazole isoten hybrids. And these hybrids were also obtained in good to excellent yields. These reactions were straightforward and there was no problem. So here's the activity table. If you see the glucosidase inhibition, the IC50 values, the activity of the compound number 10 was this compound number 10 was found to be the most active compound, having the IC50 values 0.98. So this was, if you compare this compound with the standard, this is highly potent compound. So our next target was to get the simple, use the carbonyl reactivity and to produce simple Schiff-base reactions. We treated this phenoxyphenate isotene with different substituted anilines to produce these Schiff bases of the phenoxyphenate isoteins. These Schiff bases were further reacted with thioglycolic acid in toluene solvent under flux conditions for 8 to 12 hours to produce this kind of spiro compounds. So these spiro compounds were then tested for their alpha glucosidase inhibition potential. So as you can see in this table, these compounds are also found to be highly potent if you compare these IC50 values with the standard acarbose. Then we attempted to change the substitutions at the end position of isotene. So for this, we prepared 2 different intermediates. The one is this alkyne moiety bearing the isotene, and the other one was the azide bearing moiety. So the isatin was reacted with propyl bromide to get this propyl substituted isatin, and the other one was treated with the azides. So the azides and the alkynes were coupled together by using the click chemistry in the presence of copper to get the triazole linked isatins that can name as the triazole isatin hybrids. So all these reactions were good, and we got the high yields of the products. So if you look at this table of alpha glucosidase inhibition, you will find that all of these compounds are highly active, with the most active compound have the chloro substitutions at the isatin moiety as well as the phenoxy pendant. So the next was to use the 3 carbonyls reactivity of the triazole hybrid. So the isatin was treated with the different acid hydroxides to produce hydrazones. So here is the physical data table, and you can see the yields obtained were, because this was a simple condensation reaction, so the yield was good to excellent. So again, if you look at this table, you will observe that the high potency of these compounds against alpha glucosidase. The next was to condense the triazole isatin hybrids with the thiosaminocarbazones, and in the presence of acetic acid, and we produced thiosaminocarbazones hybrids, thiosaminocarbazones triazole isatin hybrids, in good to excellent yields. So if you look at this table, these compounds are also found to be highly potent. So next was to cyclize this thiosamicarbazone moiety and produce the triazole thiazole isatin hybrids. So this molecule has 3 heterocyclic moieties in the framework. So this reaction works well, and we got the good to excellent yields of all the products. And if you look at this table of alpha glucosidase inhibition, again, the activity was exceptional if you compare the IC50 values of these compounds with the acarbose. So the purpose of preparing different series of related structures was to explore the structure activity relationship. How can we observe changes by replacing the different substitutions and different fragments in the structural framework? So if you look at this slide, so just give me 1 minute. So it is not possible to explain the detailed structure activity relationship for each table I have shown on previous slides, because due to the time limitations. Here we have presented first most active compound, second most active compound, and the least active compound from each series, just to give you an overview of the effect of different substitutions present at different positions on activity. So careful analysis of the IC50 values indicate the presence of chloro and bromo substituents at 5th position of isatin is beneficial for the activity. If you look at all these in all these series, so you will observe that the chloro and bromo substituents at 5th position of isatin is beneficial for the activity, although all the compounds are reported to have highly potent activity. So is beneficial for the activity as compared to those derivatives which have no substitution at this position. A similar beneficial effect has been observed for CLBr substitution at 4th position of the phenoxy ring. So this substitution in the yellow color, you can see this. If we have the chloro or bromo substitutions at these positions, so this is also beneficial. So overall, a slight variation was observed in the activity, but if you compare the different substituents presented as R1 and R2, so the chloro and bromo substitutions has beneficial effect. So next is the urease inhibition activity. As we all know that world population is increasing day by day and is predicted to reach over 10 billion people within the next 30 years. And source of bioavailable nitrogen are not enough to guarantee food supply for growing world population. In modern agriculture system, the use of chemicals to fertilize soils and plants is common. And nitrogen fertilizers have been widely used to improve crop productivity. Urease is an enzyme from the class of hydrolases which catalyzes the decomposition of urea into ammonia and CO2 and the loss of excess nitrogen to the environment. So one strategy to reduce ammonia emissions is the application of urease inhibitors as additives to urease-based synthetic fertilizers to block the formation of ammonia. Recently, many hybrids showing good urease inhibition motivated us to screen our compounds for urease activity. So here are the results of previously described synthesized compounds against the urease. So if you look at this table, you will observe that as compared to the positive control thiourea, our compounds, most of our compounds shows more activity, are more better. So this is the compound number 12 was found to be the most active with the IC50 values of 4.55 as compared to the 22.45 for thiourea. The next is the next scheme. If you look at these compounds, you will observe the activity of these compounds is also exceptional as compared to the standard. Same is the case here. Next, standard compound. If you compare the thiourea with our compound, most of the compounds are potent compounds. So here is the next one. Due to the time limitation, I will not go into the details. So
you have 2 more minutes.
2 more minutes. Okay. If you look at this table, this slide, so you can see the interesting structure activity relationship. Interestingly, the presence of chloro and bromo substituents at position 5 of physatin is again beneficial. However, it is hard to assign the influence of different halogen substituents at position 4 of phenoxy group. This can be attributed to many fertilizers. This can be attributed to many factors that include steric, as well as the possibility of various non-covalent interactions with the binding sites. So clear-cut structure activity relationship is hard to predict here. So however, if you compare the activity of our compounds with the standard, so our compounds are found to be highly potent. So in conclusion, I will conclude that these compounds are highly potent glucosidase inhibitors as well as urease inhibitors. Finally, I would like to acknowledge and say thanks to Higher Education Commission of Pakistan and Quaid-e-Azam University for their financial support. Thanks to the Natural and Medical Sciences Research Center, University of Nizwa, Oman, for helping us and screening our synthesized compounds for biological activity. Thanks to the organizing committee of the 4th International Conference on Applied Sciences, Mathematics and Informatics, especially Professor Stupo Hadi, for inviting me to present this keynote lecture. So I'm highly grateful. Thank you.
Okay. Thank you. Thank you very much. Very interesting topic is about isotin. Okay, we come up with the question and answer. Is anyone from the floor want you directly asking or discussing some question with Dr. Chachawan and Dr. Nazir? You can unmute your phone, please. Anyone?
Hello, Mrs.
Okay.
Moderator. I would like to ask.
Yeah, sure. Okay, Professor Sutopo, go ahead.
Okay, thank you, Mrs. Moderator, Dr. Endang Lini Mudia Tusdi. My first question, I will ask Professor Chachuan later on, but I will ask first Professor Nasir. How are you, Professor Nasir? Everything all right?
Thank you very much. I'm fine.
Thank you for your very interesting talk given to us today, and it is very inspiring for us. Yeah, I have some question to you. Yeah, the first one, for the isotin synthesized compound, it is mostly for the alpha glucosidase, right?
Yeah.
So, did you do this all synthesis in the lab and you characterize by yourself or what? That's the first question. The second, is there any anti-cancer activity for this compound? Anti-cancer.
Cancer. Yeah, because you mentioned about the anti-cancer potential about this, right?
Yeah. So first we have synthesized all these compounds in our laboratory. There are a lot of other compounds we have synthesized in our laboratory. Just presented few of the results. So all these compounds were tested against alpha glucosidase and urease. Unfortunately, at the present we cannot we couldn't study the anti-cancer properties of these compounds. So maybe we will send samples to somewhere else to have the anti-cancer studies.
Oh, so far you have never done anti-cancer activity for this compound?
Yeah, because of the lack of facilities and because in our setup we cannot screen these compounds for the cancerous properties, so we have to send our samples to other labs in collaboration.
Okay.
Yeah.
Thank you for the information. I think I'll give another participant to ask you first before I ask Professor Chachuan later on.
Okay.
Is anyone in the floor? Please raise your hand. Or at least you unmute your mix. Oh, maybe I come up with a question from the chat box before the Professor Sutopo asking some question to Dr. Chachawan. Here the question from Rido Nahrawi from the University of Lampung. He said that he's very interested in about isotin, and he want to ask how the mechanism of isotin against diabetes mellitus.
It is hard to explain the exact mechanism, but as I told you on some slides, that because of the ability of these isotins to make the hydrogen bonds, because of the presence of hydrogen bond acceptors and hydrogen bond donors, and the possibility of making the pi-pi stacking interactions, and additionally they have the possibility of making carbonyl-carbonyl interactions. So owing to these interactions, it has the potential to interact with the binding sites for the inhibition. So at the moment, we are not clear about the mechanism, but we believe that the pi stacking interactions of these the multis plays a very important role in the biological activity.
Okay.
Yeah.
I have an idea, but one question actually, not an idea. Is there any natural resources that you can get the isotin from natural resources, like from plant or from?
Yeah, because this is a natural product, so you can have this from the natural resources. But this now presently this is commercially available. This is a cheap, inexpensive material, so you can easily buy from the market.
How cheap is it, Professor Nasir? How cheap is it?
This one,
yeah,
I think you can buy 100 grams of this about $25 to $30.
Oh, very cheap.
Quite cheap, yeah.
Yeah, 1 gram.
Yeah, 100 grams.
100
grams.
100 gram. 100 gram for
cheap.
$25.
Yeah, $25 to $30. So simple, and you can also have the substituted isotins, especially the substitutions at the aryl ring of the isotin. So yeah, those are a little bit expensive, but not that expensive.
Okay, I'll continue my question actually.
Yeah.
For the substitute isotin, is it better in activity for alpha glucosidase or which one? And whatever, or what happened with that one? With the substitute isotin.
Okay, can you please repeat the question? I cannot hear.
For the substitute isotin, will it be better in alpha glucosidase activity compared to the original isotin?
Yeah, so we found that the substitution at the fifth position of isotin, when we took the halo substituted isotins, those were found to be more potent as compared to the simple isotin. So chloro and the bromo substitution, especially on the aryl ring of the isotin, was found favorable.
Okay.
How many times the activity?
Not that much, but because all our compounds are highly potent, there was a little bit different. So if you compare this unsubstituted isotin with the chloro and bromo substituted isotins, you can see that maybe 2 to 3 times more active as compared to the.
Okay, excellent then.
Yeah.
Okay, Mrs. Moderator, can I ask question to Professor Chachawan?
Yes, please.
Okay, thank you.
He's ready to answer your question.
Sorry,
right, Dr. Chachawan?
Because sorry, Professor Chachawan, because Professor Nasir is chemist just like me, so I focus on his presentation first. Okay, it is very interesting because you develop resistant rice, and Thailand and Indonesia rice is the most the main food for daily life, right? Okay, my question is, I'm sure that you have done many research for rice, right? So far, the result that you have got in the laboratory, how strong compared to the normal rice? Because I missed that one a bit when you give presentation, because I was on the way at that time. Please explain about that one.
Okay, the resistant rice variety that we identify from the Thai germplasms is named JHN or Jao Hom Nin. And this rice is actually can show like more than 80% resistance to the
80%.
Yes.
Very good.
To the collections of rice blast isolate in Thailand. It means that if we collect hundreds isolates from different places in Thailand, and then we check the pathogenicity, about 80% of the isolate cannot cause the disease in this rice.
Oh, that's very good then.
Yes, it's very good. But when we identify the resistant genes, we found that it is the PIK and PISH, and somehow the allele of the PIK that this rice has is not that strong allele. It's the PIK that can recognize only one allele of the ABR PIK from the fungus. So we still have the opportunities to improve the resistance of this rice.
Okay. Maybe I just following the question.
I haven't finished. Sorry.
Okay. Sure. Go ahead.
Sorry. But it is interesting. Why? Because rice is the staple for Indonesia and Thailand, right? Was staple for Indonesia and Thailand. So for the rice that you have developed, has it been used widely in the community?
Yes.
Or just still in the lab?
Yes. We collaborate with the breeders.
Yeah.
So once we identify the resistant genes, then we develop the DNA markers that our colleague who are the breeders can use those DNA markers to screen and integrate these resistant genes to the commercial rice varieties. Because Homnin rice is actually, the yield is not that good and the quality is not as good as the jasmine rice, but it's just show the broad resistance. So that we work together with the breeders so that they can integrate these good resistant genes into the backgrounds of the jasmine rice. And we actually, these past 5 years, we released several rice varieties that still have a good qualities of the original backgrounds with the integrations of the rice blast disease resistance.
Okay. Thank you, Professor Chachuan. Very nice and very inspiring talk for us. Sure.
Yeah. Okay.
I have one more question for Professor Nasir, Mrs. Moderator. Can I ask?
Go ahead. Yeah, sure.
Okay.
We still have. We still have 10 minutes to go.
Okay. Professor Nasir, sorry. For the isotin compound, do you think which one will be better to be used in DM type 1 or DM type 2?
Because hello,
yeah, hello,
can you hear me?
Yeah, I'm listening.
I think it's it is hard to treat the type 1, so usually we treat we target the type 2 diabetes. So we just use the different strategies to control this diabetes. So one of the strategies is to inhibit alpha glucosidase. So we targeted alpha glucosidase.
Okay. Have you ever compared with inorganic compound such as chrome compound, something like that?
No, not yet. But if you look at the structure of the isotin and our derivative, so you will see that this isotin moiety has also the possibility of coordination. So it is a kind of bidentate ligand. So you can prepare different metal complexes. So because we are the organic chemists, so we are not trying the complexes, but in case we have some collaboration with other colleagues, inorganic colleagues, so we can make the complexes of these compounds as well.
Can we have discussion about the one later on? Because I'm a coordination chemist, I'm organometallic.
Yeah. Why not? Why not?
I'm very interested with that one. It is very interesting. I'm interested in it.
If you like, we can send at least we can prepare one ligand in a large quantity, and we can send you, and you can test that.
Let us try in our lab then, because my colleagues also doing the sort of research, and we found some interesting result for the chrome compound.
Yeah.
Okay, thank you, Prof. Nasir and Prof. Chachuan. Very nice and very inspiring.
Thank you very much.
Thank you for your coming for this conference. Thank you so much.
Professor Hadim, maybe if Dr. Nasir send you isotin, maybe you can also try for the cytotoxic for
anti-cancer. Yes,
in vitro.
Yes, correct.
In vitro by using the HeLa cell cancer or something like that.
Sure, surely.
You can join with me, okay?
Yes, okay, excellent. Yes.
Thank you so much.
Professor Nasir. Dr. Widyastuti also agreed to use your compound for the HeLa compound. I'm sorry, HeLa cancer.
It will be my pleasure.
Okay, thank you.
Thank you so much.
Thank you so much.
I have one question for Dr. Chachuan. Is it possible for you to generate some, like for example, enrichment of the rice by using the fungi, whatever by using the genetic diversity, by enriching the vitamin A, for example, in the rice? Because we know that the rice is contained out of vitamin B, but we also need the vitamin A. So maybe you can combine or something like that, so you can enriching the rice with the vitamin A. Is that possible?
Yes, of course. You probably heard about the golden rice before,
Yeah.
right? That's basically what you're talking about. But the major problem with that, in at least in Thailand and several countries in Southeast Asia, is the law, because the law doesn't allow us to grow the transgenic rice in the field yet.
Okay.
So we have the ability to produce those kind of rice varieties, but we cannot release it for the farmers to grow yet.
Okay.
So that's the
So it's
problem.
still, but that the golden rice is already in the sale, right? In the United
Yes.
States.
Yes.
One of the funding is a friend of us, is Professor, I forgot her name, but he's an Indonesian that teaching in the Louisiana University. Right, it's very good. Anyone else want to asking some question? We still have, hold on for a second. Okay. We still have 2 more minutes for question and answer. Anyone? Bu Rosma mau nanya?
Maybe Probasinton maybe.
Oh, well, Papa Sinton? Is he here? Okay. Okay. Bu Rosma?
Okay.
Yeah.
Thank you. Oh, Endang.
Yeah.
It's so interesting about that your presentation, Dr. Kachwan, Janta Surya. Okay. The problem is in the development of resistance in Indonesia usually because so many like the variety of the disease. So I thought in Thailand, Indonesia is also look like the same. So it's hard to release one of the variety to the market. So my question is, do you have already successful to the release to the market one of your research? Thank you.
Okay, thank you. Thank you for the questions. And I totally agree with you that it is very difficult to identify like what type of resistant genes that we should use to develop the resistant rice varieties, because there are so many resistant genes. So the information that we need to have is the natural varieties of the fungus or the pathogens in the fields, that we have to have the information of the fungus in order to design that which resistant genes that we want to use. For example, in my presentations, I present that PIK is work really well in Thailand, but this PIK is absolutely nothing for other countries, because the fungal population in different locations or different countries are totally different. So we need to have the information about the AVR genes or the effector genes from the fungus in order to identify the resistant genes that suitable to be used for the developing of the resistant rice. And in our case, we have been make a backcross breeding program to develop the rice varieties that has the good cooking qualities together with the resistant genes, and has been released out to the farmers. They are quite a challenge for us to try to convince the farmers to accept our new rice varieties. But the best way that we have tried is just to show them by growing like side by side field with our rice and their preferred rice together. And once the natural infection of this disease occur, they can see by their own eyes that our rice varieties perform much better than the previous rice variety that they used before. And with this, we don't have to convince them anymore, because they can decide by themselves that if they want to grow their own rice, then they will not get as much yield as they want. But with the new rice varieties, it can tolerate or get less level of the disease. And we have been very successful by doing this, showing case, comparing the old varieties with the new varieties. And then another problem that we have with the commercial level is the name of the varieties, which is very, very important. Even though we tell them that this is actually exactly the same genetic information of the jasmine rice, for example, but just integrate one resistant genes into that background. And then when we change the name or even add something after the name of jasmine rice, then the price of that new varieties, it will not be high as high as the original jasmine rice, just because the brand that maybe the business company is trying to force us to not like having high yield of the high price rice varieties, but we will keep trying to help our farmers so that they can gain the benefit from the new rice variety as much as possible.
Okay, thank you. The problem also in Indonesia is that the company, which is they have their own label with all the product from our lab. Sometimes you have to challenge them. Okay, thank you. Congratulations.
Thank you very much.
Thank you. This very interesting topic for first session of the keynote speaker. Once you can have a lot of rice, but don't worry about diabetic because there is an isotin, right? So let us give applause to two very, very interesting speaker for today's session. Thank you very much, but Dr. Chachawan and also Dr. Nazir, the committee would like to give you some certificate for both of you. For first one is for Dr. Chachawan Janta Surijarat. Okay, thank you very much. We're going to send you through the email. And the second one is for Dr. Mozam Nazir. Committee?
Where is it?
They said they prepare it. Wait for a minute.
Okay. No problem. Anyway, thank you so much.
Okay, hold on for a second, Dr. Supotobo, not yet. Okay, this one is certificate for you, Dr. Muhammad Mozam Nazir. Thank you very much for your talk. Thank you very much for Dr. Chachawan also. Thank you very much for Professor Sutopo, Professor Rosma Hasibuan, and also for other audience. Thank you very much. I return back to the MC. Thank you.
Thank you.
Thank you.
All right, thank you very much Bu Endang Linirin Widasuti PhD for guiding us strictly in the first plenary session.
Next plenary session will be guided by Dr. Renat Ronius Marjunus. But before that, let me read his brief curriculum vitae. His full name is Dr. Renat Ronius Marjunus, SSI, MSI. He is a Vice Dean of Student and Alumni Affairs, Faculty of Mathematics and Natural Science, University of Lampung. His educational background: Bachelor, Gadjah Mada University Department of Physics in 1999. Master, Gadjah Mada University Department of Mathematics and Natural Science 2002. Doctoral, Universitat Derbansyah Munchen Institute for Physics 2018. There are 37 of his publications. in the past year. One of them is in 2020, processing of granite quartz solid waste into industrial high silica material using leaching process with HCI concentration variation. Please welcome our second moderator,
Dr. Renat Ronius Marjunus.
Yeah, thank you very much for the MC. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good morning, Tabi Pun.
Ya ampun.
So the Tabi Pun is the special or the local greeting from the Lampung province. Means that please excuse me to give a speech. So the answer is please go on. Ya Pun. So ladies and gentlemen, this is a very nice opportunity for me to be a moderator for our third speaker. Before the speaker gives the presentation, I would like to introduce our speaker to all of the audience in this conference. The next speakers will be presented by Associate Professor Dr. Adibah Suib. The title of her presentation is Optimization Models for Collection Routing Problems in Municipal Solid Waste Management. Associate Professor Adibah Suib, now she is as a Deputy Director from the Malaysia Institute of Transport, University Teknologi Mara, Shah Alam, Selangor, Malaysia. Her academic qualification, she graduated from the Bachelor of Mathematics at the Old Dominion University in Norfolk, Virginia, USA. And then she graduated also from the same university for her master degree. And then finally, she got her PhD in mathematics and statistics at the University of Birmingham, United Kingdom. There are many achievements that have been done by the Associate Professor Dr. Adibah Suib. For instance, she got the gold medal at the Seoul International Invention Fair in 2009. And then she also has done many research. So the last one was vehicle routing problem with the time windows based optimization models for sustainable grocery delivery using homogeneous and heterogeneous fleet of vehicles. She got this grant from Malaysia government, and then there are still many achievements that is not possible for us to explain in this time. So finally, I would like to invite our speaker today, Associate Professor Dr. Adibah Suib. So the time is yours for something like 20 minutes, please.
Okay, Bismillahirrahmanirrahim. Thank you to the moderator for the kind introduction. Assalamualaikum, selamat sejahtera, and good morning, to Honorable Top Management of University of Lampung, Top Management of the Faculty of Mathematics and Natural Sciences of UNILA, and also the organizing committee, professors and lecturers, students, and all participants of this conference. First of all, I would like to say thank you so much, to the organizing committee for inviting me and to share some insights on the research. Yeah. Okay. Let me just share the slides. Yeah. Can you see the slide?
Yes, Prof.
Okay. All right. So today my talk will be on the optimization models for collection routing problems in municipal solid waste management. Okay. So this presentation will contain the overview of MSWM and also two corresponding research, yeah, and also the conclusion. So basically, yeah, the interest on this research is based on the issue of waste collection and transportation costs, which accounts for a large bulk of the total MSWM or municipal solid waste management, which is about 75%. There's even slight enhancements or improvements in the operations, especially in the collection and routing, will actually significantly reduce the cost. So the study is concerned with two research, yeah, which is on the mixed integer goal programming model for two cities, namely Shah Alam and also Kuala Lumpur. One is on the transportation problem and the other one is on transshipment. So results indicated that for Shah Alam, which doesn't have transportation at the time of the study, we are able to propose a location or site for the transportation. And as for Kuala Lumpur, there is a reduction in terms of amount of waste brought to landfills. So let me just present to you an overview of MSWM and also the phenomena of increasing waste generation worldwide. So as we know that we have the issue of urbanization, yeah, coupled with the rapid economic growth and lifestyle changes, these contributes towards the increase in waste generation. So globally, there is an increase of or projection of increase from 2.24 billion tons of solid waste in 2020 to more than 3 billion tons in 2050. Yeah, so that has been a great concern to all cities globally, yeah, including Malaysia. So in Malaysia itself, the waste generation accounts for 38,427 metric tons, where still 85% of this waste go straight to landfills, yeah, and the recycling accounts for just small amount of this total waste. So issues of solid waste management has been given special attention by the United Nations. So this can be seen from the targets of the Sustainable Development Goals, namely SDG 11 and SDG 12, target 11.6, target 12.4, and target 12.6. Yeah, sorry, target 12.5. Yeah. So in this case, what we can see that the United Nations has actually emphasized the importance of solid waste management, namely the municipal solid waste, through the World Habitat Day 2022, yeah, asking for communities and cities and towns around the world to increase innovation in handling the waste management. Yeah. Okay. So for this part presentation, the focus is on the collection and transportation, which is actually about 60 to 70, which I mentioned just now, can be up to 75% of the total cost of the municipal solid waste management. Okay, these are the sources and types of municipal solid waste. Yeah, namely, we can see that the municipal solid waste actually covers the processes of collection, transportation, treatment, and processing of waste until the disposal of the solid waste to the at the landfills. Yeah. Okay. So for these studies that I'm going to present, it covers only residential, commercial, institutionals, yeah, waste, solid waste only. Okay. At the same time, globally we are also into circular economy, which means that the waste needs to go through to the processes like 3 R's or 5 R's, reduce, reuse, recycle, remake, or repair, for examples, in order for us to reduce or minimize the waste that goes to landfill, at the same time to generate income from waste. So let us look at the applications of mathematics and operation research in the municipal solid waste management. As you can see that, at the point of collections, at the residential areas and so on, we can have smart waste collection system where the bins are equipped with sensors to indicate when it's filled and the time for the best collection. And then we can also look into facility location allocation problems, where we need to determine the sites for the facilities like transfer stations and also the landfills. And of course the vehicle routing problem or arc routing problem, and similar, regardless of whether we are using the GIS incorporated data for the routing optimization. So in other words, there are a lot of studies can be done through collection routing optimization. And of course, at the landfills, there can also be mathematical models on the leachate production. So namely, these cover the strategic, tactical, and also operational level. So let me first start with the first research, which is on the mixed integer programming model for transportation problem, a case study of Shah Alam done by my student Nur Hayatul Asna. Here, the concerns is on the municipal solid waste management in Shah Alam area, which comprises of 44 collection zones and 56 sections, I think. In this study, mixed integer programming is formulated. And at the moment, at the time of the study, there has been no transfer stations in Shah Alam. So therefore, there are 3 potential transfer stations proposed. And the research covers on the background of study, literature review methodology, results and discussion, and also conclusion. And here, we can see that, for example, as the background, based on Young et al., 2019, 89% of collected waste in Malaysia were not treated fully and ends up in the landfills. And then for Selangor, Jamil 2018 says that Selangor is the highest ranked states, compared to other states in Malaysia, with highest rank of waste, in terms of daily solid waste production in Malaysia. Okay, so that is the reason why we have chosen Shah Alam as an area of study. So for this study, it just concerns with transportation from collection areas to the transfer stations. So this is a transportation problem as compared to the next research, which is on the transshipment problem, where it involves transportation from the collection areas to transfer station and then sent to the landfills. Okay, so in terms of vehicles, we are using heterogeneous valid vehicles, which include compactor and also open tipper, which have very different capacities. So compactor is 10 tons and open tipper is 5 tons. Okay, and then as mentioned, 3 transfer stations proposed. Okay, and here we are going to formulate the problem as mixed integer programming, but with the presence of multiple objective functions, we have the goal programming. And as for the solution method, we are using MATLAB, which actually include heuristics and also exact method, in the solver. So for literature review, we have covered comprehensive reviews, but we concentrated also on the 8 selected past studies which have mathematical programming models which will be similar to the one that we are targeting. Okay. And we carried out gap analysis in terms of the objective functions, decision variables, and constraints, and so on. Okay, so this will set the basis for the main characteristics or features of our mathematical model. Okay, so this is a summary of the gap analysis. That is why we have incorporated new objective function in the model, and we also consider heterogeneous vehicles, and we use MATLAB for the solution method. Okay, this is the research flow chart, and also the phases of the study. And this is actually the map of the collection areas that are involved. 58, sorry, I just now mentioned 56, 58 sections with 24 zones, collection zones, which are involved, and these are data that have been collected, that covers in terms of the amount of waste generated and the usages of the vehicles. And for the study, we carry out modification on some reference models, 3 reference models as mentioned here, namely, and these include, we actually come up with new notations and also add new objective function and respective constraints as emphasized by the service providers at the Shah Alam city. Okay, so as for result of objective 1, this is actually the analysis of data. The summary is written here. So this is the amount of waste generated, the maximum and the minimum, and also the shortest path and also longest path at the respective collection to transfer station, etc. Okay. Next, the second objective is to come up with a modified objective function and constraints, to actually complete the formulation of the mixed integer programming model. So these are the indices, decision variables, objective functions, sets, and parameters introduced, in this study. And here is the complete formulation, with the 3 objective functions, namely minimize total transportation cost, carbon dioxide emission, and noise pollution, and subject to various constraints emphasized by the service providers. And we have next the result for the objective 3, where in this case we are going to solve the models for optimal solution. So in this case, the activities done is to prepare the input data in Excel, and then transfer the matrices into MATLAB correspondingly. Okay. And as for result of goal 1 or objective function 1, basically, since we are using the preemptive goal programming model, the objective function are actually arranged according to priority. So the first priority A or goal 1 is to minimize the total transportation cost. We have this per collection. The amount is 2,320 ringgit Malaysia. And the result shows that most of the waste are sent to the nearest transfer station. Of course, this is to minimize the total distance, so that we can come up with the lower cost, minimum cost. And then the next one is the result for objective 2, which is on the carbon dioxide emission, where the minimum transportation cost is whole for 2,320.24, and we have this 1,287.22 tons as for the solution for goal number 2. Okay, so well, in terms of the examples of results here. For example, here we can see that goal 2, we can see 15,000 tons, and we can see also the 5.37 tons. This is the total transportation, also the carbon dioxide emission. And then we have result for goal 3, which means here to minimize the noise pollution. Yeah. Okay. And based on the observation, we can see that selection of transportation and volume of waste transported remains the same as in goal 2. So the first two remains the same because these are the constraints, additional constraints enforced into the model after we carried out iteration number 1 and iteration number 2. Okay. And next, we have the result for objective 4. This is the complete strategies that can be formulated based on the solution of the model. Yeah. So based on the 3 potential transfer station, it seems that the Nai Alam is the best choice because it receives the most tons of waste, yeah, as compared to the other 2, namely Meru and Kota Kemuning. Yeah. So these results also summarize the types, number of vehicles and types of vehicles used for transporting the waste from the collection areas to the transfer stations. Okay, so if based on the result, you can see that even though we have 95, yeah, open tippers, yeah, the model has been able to reduce the number of vehicles used or open tippers used to 74 rather than using or fully utilizing 95 available vehicles. And in terms of compactor, 38 out of 39 compactor is used. Yeah. So in other words, the model is able to reduce the number of vehicles used. Okay. So from these, actually, the study has been able to propose the best location for the transfer station, namely the Nai Alam, and also all the objectives of the study have been achieved. Okay, that's for the first study, and these are some of the references. Yeah, and then let's compare this to the other type of study, which is the preemptive mixed integer Go programming for transshipment. Yeah, as I mentioned, transshipment means you will be involved with transporting, yeah, from the waste, yeah, from collection areas to transfer station, and then the next stage is from the transfer station to the landfills. Yeah. So this study is carried out by Siti Khairunisa binti Muhammad Roslan. Yeah. Okay, and then we have here, yeah, the contents. Okay. First of all, yeah, again, the concern is to be able to efficiently carry out the municipal solid waste management. Okay. This study concerns with the municipal solid waste management in Kuala Lumpur. If you look at the compositions of waste for Kuala Lumpur, the main bulk is on food or organic waste, followed by plastic waste, and then paper, yeah, papers. Okay. All right. So the idea is to fill the research gaps where there are, yeah, there has been lacking in studies in terms of, yeah, handling municipal solid waste management by using the mathematical programming model and treating it as two-stage transportation or transshipment model. Okay, so these are the research objectives of the study. Yeah. Okay, and for Kuala Lumpur, yeah, Federal Territory, we have 17 schemes. These are the collection zones and 2 transfer stations. As compared to Shah Alam, Kuala Lumpur already have transfer stations. Yeah, so they already have transfer stations where here there are certain processing or certain efforts to reduce the waste. Yeah, and then we have 2 transfer stations and 3 landfills. Yeah, so stage 1 is transportation from collection zones to transfer station, and next followed by the transporting waste from transfer stations to landfills. Okay, so data has been collected, and this map is showing the locations of the transfer stations. Yeah, the transfer stations. Basically, I forgot to mention, 2 transfer stations already exist. They want to add another transfer stations. Yeah, so that is the idea. Okay, and currently there are 2 landfills. Yeah. And the landfills are located outside of Kuala Lumpur. Okay, so here we see the structure of the transshipment. As you can see that we have from schemes, yeah, collection schemes to transfer station to the sanitary landfills. Okay, now, so basically the main novelty is on the formulation of the transshipment or two-stage model using MIGP. And of course, the significance is to the municipal solid waste collection service providers in Kuala Lumpur. Okay, so some literature review or past studies, you can see, yeah, some studies or related past studies in Australia, Iran, and Palestine. And we also studies in Tanzania, Argentina, and also in Malaysia. Okay, next. This study involved 7 main steps or research activities, yeah. And as I mentioned, that the models will be solved using the MATLAB optimization solver called InLinProc. Okay, let us look at the results and discussion. Yeah, so recall that there are 4 objectives of these studies. First is to analyze data collected from Kuala Lumpur Municipal Solid Waste Management. Okay, so in this case, you can see that the amount of waste, total distance, yeah, to transfer stations, and also number of compactors and number of open tippers used, yeah, in this case, utilizations of vehicles, and also that is actually for stage 1 and also for stage 2 from the transfer stations to the landfills, yeah. So there are landfills here, yeah, and then okay, the next objective is to be able to formulate the mixed integer programming with multiple objectives that will be solved using the goal programming method with 3 objective functions. Okay, namely, we have the same objective functions in stage 1 and stage 2, where we are aiming to maximize the waste collection and transportation costs, and also minimize the carbon dioxide emission. The same also applied in the stage 2 of the model. Okay, so in this case, we can see the constraints involved, yeah, for stage 1, yeah, in terms of capacity of vehicles, yeah, selection, yeah, and also the maximum load, yeah. And then for stage 2, we have additional constraints, yeah, besides the implemented additional constraints due to the preemptive goal programming method. Okay, next is to solve the formulated model, yeah, using the preemptive goal programming method. So you can see that for this, yeah, there are 988 decision variables, yeah, for stage 1, yeah, and also, sorry, yeah, let me just yeah, look at this, and there are 12 decision variable for the stage 2. So in this case, what we can have, or we can see, this is the stage where we actually prepare all the matrices involved, yeah, for execution in MATLAB. Yeah, so that's where we actually expand the models first, and then from there, all these matrices are actually imported to MATLAB and solved using InLinProc. Okay. Okay, then for stage 2, we also use MATLAB. And the last one, or the last objective, is to come up with strategies for the municipal solid waste management for Kuala Lumpur. Okay, let's look at here. Yeah, from the 150 vehicles, yeah, available, yeah, the number of vehicles used is reduced to 144. Yeah, and then here we have For open tipper, this is compactor of 10, 15, and 20 ton capacity, and open tipper 3 tons, 5 tons, and 8 tons. Yeah, so these are heterogeneous vehicles. The total number of vehicles utilized is only 27 as compared to available 46. And for volume of waste, for collection areas to transfer station 1, Taman Beringin, 1,144 tons. And we have 1,155 tons for Kampung Bohol. Together, a total of 2,300. And for the stage 2, we have prime mover, yeah, being used. This is commonly done, yeah, when transporting waste from transfer station to landfill. We are using larger vehicles, which is prime mover, of capacity 30 tons. Yeah, there are 65 units available. Okay, and in this case, there are 43 prime movers are being used. Yeah, from the 65 available. Okay, and volume of waste is 1,264. Okay, so if we look at the summary, yeah, we can see the total transportation costs from stage 1 and stage 2 total up to ringgit Malaysia 3,164, yeah, per collection. And in terms of carbon dioxide emissions, we have 124.77 tons, and total waste collected has been reduced. Yeah, if we look at 2,300, and then stage 2 we have 1,264, and finally the last one to be brought to landfill is only 1,335 and 54 tons. That is reduced by over 40%. So as a conclusion, we can see that we are able to reduce, yeah, the transportation costs by reducing the number of vehicles used, and also the carbon dioxide emissions and the total amount waste sent to landfill. Okay, of course, these can be applied to other municipalities, yeah, or cities and towns, yeah, where for further studies, there can be other objective functions to be included in the mathematical programming model. Okay, these are selected references. As overall conclusion, we can see that by implementing efficient way of municipal solid waste management, we are going to be able to actually meet the target set for the Sustainable Development Goals, SDGs, yeah, namely SDG 11 and also SDG 12, yeah, and also to be able to comply with the yeah, calls by UN, especially for UN Habitat, World Habitat, yeah, effort, yeah, that is to be innovative in terms of the waste management, collection and recycling, and so on. Okay, so this presentation actually focus on the routing, yeah, collection routing optimization for municipal solid waste management. Yeah. As a conclusion, we know that logistic performance in the municipal solid waste management are influenced by collection and routing. And through these operations, efficiencies, the cost of municipal solid waste management can be reduced further. With that, I thank you.
Thank you very much, Professor Adiba. So it was a very nice presentation. So ladies and gentlemen, please, now we are in the discussion, the time for discussion. Oh yeah, I would like also to inform all of you, unfortunately, our second speaker from Russia cannot join with us. So it means that we still have very much time to discuss with Professor Adiba regarding her presentation. So please raise your hand if you would like to ask to Professor Adiba. Okay, there is a raised hand from from Dr. Bambang Irawan. So please, Pak Bambang.
Okay, thank you, Mr. Ronius, for the time. Prof. Adiba, I don't understand about the models, but my concern is about organic municipal waste. I see in your chart, the food or organic is almost 50%. My question is it all this type of municipal waste, the organic, also sent to the landfill, or they are treatment differently as a compost or something like that? Because it is done like that, probably it will change the model of transportation. Okay, thank you.
Okay. Let me just find the slides that actually talk about the composition. I think this is the one that you mentioned. Okay, so the main composition of the municipal solid waste is basically food, organic, and so on. Actually, the way the waste is treated differs from countries to countries. Yeah. So, for example, yeah, transfer stations, or we can also have processing plants or treatment plants. Yeah. So we can have transfer stations for segregation of waste. Yeah, segregation of waste. So we are able to come up with a certain reduction efforts. Yeah, for example, segregation means you can actually separate all the recyclable. Yeah, yeah, for the 3 R's. Yeah, and at the same time, they can be also compost. These actually the idea of transforming the food or organic waste into something that can be commercialized. Yeah, so that can be transformed, for example, to become compost fertilizers. Yeah, so that is part of the processing plants. Yeah, that can be also located at the transfer stations. Yeah, so that is a way. And then basically at the transfer station, you can have a complete treatments of everything. Yeah, so we can have the segregation and also further reduction to compost and other types of efforts in minimizing the waste. But even if we send it at the landfills, we can also have decompositions of the waste through system of layers, where actually we let the waste to settle down to transform. So in other words, to be able to settle to decompositions, where these wastes are actually we know that at landfills they have all these solids being put into an area where they dig and put the waste there. Yeah, and with the system of layers, layer system here, they can be able to reduce further or decompose this waste even at the landfills. I hope that answers your question.
Yeah, okay, thank you. It is interesting because I thought if there is no segregation and all the garbage is sent to the landfill, it becomes new problem, of course.
Yeah.
Okay.
Originally, that was
Thank
the what happened. Yes, sent straight to the open landfill. Open landfill, not the sanitary one. Sanitary ones,
not
they
sanitary.
cover. Yeah.
Okay. Okay, thank you, Mr. Matya.
Okay, welcome.
Okay.
Thank you, moderator.
Yeah, you're welcome. So the next question, please. Anyone? So maybe I would like to ask some questions. So I would like the first question to Professor Adiba. I still don't understand how can we make a model for the waste management using the as we can see in your presentations that you use. There are many sigma for this model, right?
Okay.
So, yeah, because I'm not a mathematician, so please could you please explain a little bit how can we model this problem with this this process?
Okay.
Yeah.
First of all, if you look at this the formulation of the model, basically when modeling, because there have been earlier studies for this type of problem. So what we do here is actually modification and enhancement. So as I mentioned, there are 3 reference models have been used for the first research, for example, here. So you can see, let me just mention about the transportation cost. Yeah, so here X, I, J, K, L is actually the binary decision variables. For arc routing, we actually looking into where are the collection zones and sent to which transfer station. This one is transportation problem from collection zone to transfer station. So basically, we just look at the possibility of sending from this collection zone to this transfer station. So this is binary variable indicating where or whether you actually transporting from this place to another to the transfer station and to which transfer station. And here this one here, since we have the transcript IJKL, so the summation basically this is going to actually give you all the permutation possible. Yeah, of course, I and J shouldn't be equal. So I to J, definitely I cannot be equal to J. So basically, this is providing all the permutation possible. Yeah, for you. Yeah, to have to send all this. Yeah, and then here the C is actually the transportation cost that includes the fuel consumptions and also the distance traveled by the vehicles from collection node I to transportation J. Okay, so that is for the transportation cost. As for the carbon dioxide emissions, so we have this is to show whether we have the from I, J and vehicle K, and then we have these types of vehicle. Because when we are using heterogeneous vehicles, again, if K here is the different types of vehicle, L is the capacity of the vehicle, so different capacity. And then here this is the carbon dioxide emission. We use formula by previous study in calculating the carbon dioxide emission when traveling from particular node I to node J. So node I is the collection station, J is the transfer station. Okay. And actually there has been few studies, but for this study we concentrate on noise pollution. But some people concentrate on the smell pollution. That can be done also. So this one is for noise pollution. Because especially in cities, we don't like very noisy waste or garbage trucks moving around. Okay, so this is the coefficient in terms of decibel of noises generated by vehicles type K and with the certain capacity. With a certain capacity. So that's how we actually calculate the total for all these objective functions. And the constraints are related to all these three objective functions. So we ensure the number of vehicles satisfied. We don't exceed the capacity of the transfer station. Because transfer station also have capacity in terms of waste to be sent there. And then distribution of waste. And also total collection, load of collection vehicles. Each vehicle also has capacity, so that cannot be exceeded. And then we also look at various utilization of the vehicles, and we put here budget expenditure, so it shouldn't exceed the budget allocated by the municipality or the city council.
Okay. I see. Okay. And then how does it take time for the iteration for all of this optimization?
Okay. This one doesn't take long because this even though I have here I forgot. We have 24 zones. 24 zones. So basically, if you think about this, 24 zones is not considered as large or big data. We have 24 zones. So this is actually not taking that long. I think in terms of execution, it just takes few minutes to complete the computation using MATLAB optimization solver. I'm so sorry that I don't have the number of minutes, but it's not taking that long. Not taking that long.
Okay.
Because
So
as compared to if you are using heuristics, maybe just heuristic per se, maybe not so systematic, that may be taking longer time, yeah, or maybe if you use dynamic programming or exact methods, yeah, that will be taking long. But this one here is using MATLAB optimization solver, which is quite efficient in terms of the computation.
So I thought that it takes, let's say, 1 day.
Oh, no.
Programs for.
Yeah, it's quite fast to get the answer. Yeah.
Okay.
And
So
it also depends on the types of computer that you use. Yeah. Sometimes a powerful computer can have the execution to be faster.
Okay, so maybe there are some questions from the audience before I continue with my next questions. So please, ladies and gentlemen. Not yet. So did you use also the special functions from the toolbox of MATLAB, or did you make it all by yourself? I mean, program. The toolbox.
Basically, this one here, yeah. Normally for master students, we just expect that the students of using MATLAB solver, which is already there.
Okay.
So you just have the difficulties is to come up with this formulation correctly. Basically, if you formulated the model correctly, then you will obtain the correct expansions of the model in terms of, in MATLAB, you need the matrices for the objective function coefficients, the coefficient of the technology matrices, right-hand sides, and so on. Yeah, for the inequality or equality constraints. Basically, if your model formulation is correct, the matrices will be actually established nicely, correctly, and that can be imported to MATLAB, and the execution will actually bring you to the optimal solutions, yeah, most of the times, yeah.
Oh, okay. And then if we see the result of this simulation of this optimization, that the next question is, have you ever proposed this the result of these simulations to the government? So it means that, I mean, that perhaps the government will apply or will implement this result.
Okay, thank you for the question. Actually, all the data have been gathered from the city council. For example, for Shah Alam, it's from the Majlis Bandaraya Shah Alam. So basically, the results are being shared with the city council, yeah, or the service providers. So we propose, okay, if you come up with this, or you actually carry out this particular strategies, yeah. So if you look at the strategies that we propose, yeah. For example, here, yeah, okay. Okay, so these are the ideas that we got from the optimal solutions, yeah. So which one is the best transfer station, yeah, that can cater most ways, and so on, followed by which transfer station, and the how these can be arranged in terms of the number or utilization of vehicles from collection zones to certain transfer station, and so on. So these results have been shared with the authorities or the service providers. And that is within the city council of Shah Alam. Yeah. So we actually recommended or actually proposed that there can be certain reductions in terms of number of vehicles, that can be utilized. The same goes with Kuala Lumpur problem, where actually we are able to show that currently the results here. So if we look at the results here, the number of vehicles used can be reduced further. Instead of using all 150, we can use only 144 vehicles, or in this case compactor. Therefore, if you use reduced number of vehicles, less vehicles, then reduce in terms of fuel consumptions, yeah, manpowers, yeah, manpowers use, and also we can optimize the number of tons being brought by these vehicles. Yeah. So that is actually the savings. Yeah. And then we can see the total waste. Yeah. In terms of Kuala Lumpur, which already have the transfer stations. Yeah. The amount of waste collected and then finally brought to the landfill can be reduced. Yeah. So that is actually the important thing, which is minimizations of the final waste sent to the landfills.
Okay. So perhaps this is the last question from me. Still from me, not from the audience. Yeah. When I was in Germany for my PhD, I saw that they use the separation for the trash. So as you mentioned before, as long as I know, according to my understanding, that you haven't used yet the separation of the trash as a parameter here, right?
Yes.
Yeah.
Actually, there is another study that actually handle in terms of recycling. Yeah, recycling. So that is how we look at the idea of facility location in terms of recycling. Yeah, recycling, so that the waste can be reduced or can be minimized at these recycling centers. Yeah. So this one is just on the collection routing optimization. So it is consider transfer station, yeah, but not looking at the amount of reduction based on the 3 R's, yeah, reduce, reuse and repurpose. Yeah.
So do you have a plan to continue this optimization? By using the 3 R's recycling process, or?
Yeah. Okay. Actually, we are proposing another study on the circular economy. Yeah, circular economy. That is in terms of how much income can be generated through the recycling efforts. Yeah, of waste. Yeah, so that is also a topic because I think many and many countries actually are looking into circular economy. Yeah, circular economy, especially for income generation that can help the lower income group, so that is the idea. Yeah.
Okay, thank you, Professor Adiba. So ladies and gentlemen, so is there anyone to ask some question? So as I mentioned before, that we still have much time, but if there is no more questions, so we will close this session. Nothing. Okay, so Professor Adiba, thank you very much for your nice presentation. So it was also a nice discussion for us. So the next is I would like to show the certificate for you as a keynote speaker. So please, the operator. Yeah. So again, Professor Adiba, thank you very much. So we give applause to Professor Adiba.
Thank you.
So finally, we are in the last time for this session. Again and again, thank you very much for all of the audience and also especially for Professor Adiba. So now the times I will give it back to our MC.
Thank you very much for Dr. Renatronius Marjunus.
All right, ladies and gentlemen, now we have lunch and pray break time. We will be started again at 1 p.m. for parallel session. So, for all of the participants may enter the room based on the instruction that we have sent. Thank you very much.